Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the A-Level Biology Practicals. This is core Practical 3. Investigate membrane properties, including the effect of alcohol and temperature on membrane permeability. In this core practical, we're going to be looking at how membranes can be made permeable by changes in the membrane properties. The example we use here is beetroot. Remember, beetroot contains vacuoles, and these vacuoles contain pigments. For example, there is betalain. These vacuoles are surrounded by membranes called tonoplasts. Also, the cells of plant materials contain a cell membrane, so there is going to be leakage of pigments from the vacuole as well as from the cytoplasm. Chemicals as well as changes in temperature affect the conditions of the membranes, making them more permeable to the substances that are contained inside. And again, this is the example, betalain. As the pigments leak out, the color of the solution surrounding the tissues is going to change, and this can be studied using a colorimeter so that we can know which conditions cause the greatest leakage or which conditions cause the greatest membrane permeability. So first I'm going to look at investigating the effect of an alcohol on membrane permeability. So set up experiments with at least 5 different concentrations of the alcohol. You will label the test tubes, of course, with the concentrations corresponding to the alcohol. Then we will use a cork borer like what we see here in order to cut out cylinders. These are going to be cylinders coming out of there. You use a knife and a ruler in order to cut similar lengths. You will wash the cylinders. Washing helps so that all the leaked betalain from the cut cells is extracted out or washed out, so that when you place those cylinders in fresh water or in fresh distilled water, there is no more leakage, and any leakage that will occur will be because the alcohol concentration has been varied. So here we'll wash the cylinders with distilled water and pat them dry using paper towels. And then you add each cylinder to a different test tube and leave it for about 20 minutes. This is for the different concentrations of the alcohol. After the 20 minutes, you're going to shake the tubes once and quickly remove the cylinders from each of the tubes using forceps. You discard the cylinders, and then you use a calibrated colorimeter at a specific wavelength of light in order to be able to measure the absorbance. To do that, you have to transfer the liquid from each of the tubes into the colorimeter cuvette. This should be cuvette, and determine the percentage transmittance. You could write transmittance here. Some people will measure the absorbance. So you'll repeat the experiment for all different concentrations of the alcohol and then calculate the mean. And again here, you're measuring absorbance, amount of light that is absorbed. So if there is greater leakage, it means there is going to be greater absorption because the color is going to be deeper or darker. And if there is less leakage, there is going to be less absorption and more transmittance. You can record the results in a suitable table. Since the independent variable is the concentration of the alcohol, we have to make sure that the variables like temperature are kept constant. We do that using a water bath. So the conclusion we see from here is, of course, the cell membrane contains lipids. Also, the tonoplast is made up of lipids because they're phospholipids. These are going to be soluble in the alcohol, and as the alcohol concentration is increased, membrane permeability is going to increase, and that is going to lead to greater leakage, as we can see from this graph. Increasing the alcohol concentration is going to cause greater membrane permeability, so more betalain is going to leak out, and the transmittance is going to be less, or the absorbance is going to be greater, depending on the concentration. Next, we're investigating the effect of temperature on membrane permeability. Here we're going to do the same thing, but the difference is the independent variable is temperature. So everything else is going to be kept constant, except we're setting up experiments using the same cylinder. Of course, we're going to use the cork borer to cut out some cylinders from the beetroot. We'll use a knife and white tile in order to trim to create cylinders that have the same length that we saw previously. And then we'll wash them with distilled water to take away the betalain that has leaked out. Pat them dry with paper towels, and then we'll place those in specific volumes of distilled water at varied temperatures or at different water baths at different temperatures. We'll be able to see how much has leaked out. We'll use a calibrated colorimeter set at a specific wavelength of light. We'll transfer the liquid from each of the tubes into the colorimeter cuvette and determine the percentage transmission or transmittance, and you could also measure the absorbance. Repeat the same at each temperature and calculate the mean. From 0 onwards, the higher the temperature, the greater the permeability, because as temperature increases, there is more kinetic energy. The particles or the membranes or the phospholipids are going to be moving faster, and that is going to cause greater leakage. And as the temperature increases further, the proteins are going to be denatured, and that is going to lead to further increase in permeability. But below 0 degrees, we can also see there is an increase in permeability, and this is because there are going to be some ice crystals that are formed as freezing occurs, and when these are formed within the membranes, there is going to be an increase in permeability as well. So a graph like this shows the effect of temperature on membrane permeability. So in any question asking about the different chemicals or different temperature changes on membrane permeability, expect these two graphs, as well as how the procedure can be carried out. Actually, it may not be only the alcohol, they could bring some other chemical that affects the conditions of the membrane, making it more permeable to substances that are contained inside. So this brings us to the end of this core practical. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.